For nearly 700 years, historians argued over where the Black Death came from. Some said China, others said the Russian steppes. Medieval sources pointed vaguely east, but couldn't agree on specifics. The plague killed between 30 and 60% of Europe's population, reshaped civilization itself, and no one could prove where it actually started. Until 2022, a team of scientists opened graves from a forgotten cemetery in Central Asia, extracted DNA from teeth buried in 1338, and found something no one expected: the mother strain, the ancestral pathogen that gave birth to every plague lineage that followed. And it didn't come from where anyone thought. The Black Death wasn't just another disease. Between 1346 and 1353, it killed somewhere between 30 and 60% of Western Eurasia's population. Entire villages vanished. Feudal systems collapsed. The genetic structure of surviving populations was fundamentally altered. But despite its massive imprint on history, the geographic origin remained a mystery. Medieval chronicles offered only vague clues. Gabriele de Mussi, a notary who documented the famous siege of Kaffa, said the plague came from the land of darkness, a term that could mean anywhere north of the known world. Arabic sources mentioned the land of the Uzbeks and devastation in Cathay, which we now call China. Everyone agreed the plague moved from east to west. But where exactly did east begin? By the 20th century, three major hypotheses had emerged. The China hypothesis became the dominant theory, popularized by historian William McNeill in his 1976 book, Plagues and Peoples. The logic seemed sound. Chinese dynastic records documented a massive epidemic in Hebei province in 1331 that reportedly killed 90% of the population in some areas. The Mongol Empire had unified the Eurasian landmass, creating what McNeill called a microbial common market. Pathogens could travel from endemic reservoirs in the Himalayas or Yunnan to dense population centers, then westward along the Silk Road. For decades, this was the textbook answer. The steppe hypothesis suggested the plague didn't travel thousands of miles, but rather woke up in local wild rodent populations of the Pontic-Caspian steppe, modern Russia and Ukraine. According to this model, the plague was already there, dormant in the Golden Horde territories, triggered by climate anomalies or population booms in rodents. The Central Asian hypothesis was the underdog. As early as 1890, Russian scholars had noticed something strange in the Semerekie region of modern Kazakhstan and Kyrgyzstan. Nestorian Christian cemeteries with tombstones explicitly mentioning pestilence. But without biological evidence linking carved stone to bacteria, this theory remained marginalized. The breakthrough began with archaeology, specifically excavations conducted between 1885 and 1892 in the Chui Valley of northern Kyrgyzstan. This region was a vital artery of the Silk Road, home to Christian communities under the Chagatai Khanate during the Pax Mongolica. Russian excavators uncovered two cemeteries, Karadzhigach and Barana. The graves contained artifacts that told the story of a cosmopolitan trading hub: Chinese silks, Golden Horde coins, pearls from the Indian Ocean, corals from the Mediterranean. But the real revelation was the tombstones themselves. 467 tombstones spanning nearly a century, from 1248 to 1345. But here's what made researchers stop. 118 of those tombstones were concentrated in just 2 years, 1338 and 1339. 10 of them explicitly stated the cause of death in Syriac script, the liturgical language of the Nestorian Church. The word was matana, pestilence. One inscription provided chilling specificity: in the year 1649, and it was the year of the tiger in Turkic bars. This is the tomb of the believer Sanmak. He died of pestilence. This was 7 to 8 years before the plague reached the Crimean port of Kaffa, a catastrophic mortality spike in a remote Central Asian trading town, documented in stone. But here's the problem. Pestilence in the 14th century could mean anything: typhus, smallpox, dysentery, cholera. Without specific descriptions of buboes or the characteristic blackening of extremities, it was just a generic term for mass death. The inscriptions were compelling, but they weren't proof. For over a century, this remained a tantalizing clue without biological confirmation. The skeletons excavated in the 1880s had been transferred to the Peter the Great Museum of Anthropology and Ethnography in St. Petersburg, the Kunstkamera. They sat in storage, cataloged but unexamined. Then, in the mid-2010s, historian Philip Slavin conducted a comprehensive reanalysis of the original inscriptions. His demographic reconstruction confirmed what the numbers suggested. This wasn't normal mortality. This was a community in crisis. But to prove it was the plague, the one that changed the world, they needed DNA. Extracting genetic material from 700-year-old bones is nearly impossible. DNA degrades over time into short, damaged fragments. Soil bacteria and environmental contamination overwhelm everything. Even if you find pathogen DNA, you have to prove it's authentic and not modern contamination. The research team, led by Maria Spyrou and geneticist Johannes Kraus, used a technique called capture enrichment. Think of it like fishing with a highly specific net. They took samples from the pulp chambers of teeth, a skeletal niche that's vascularized and can preserve traces of blood-borne pathogens present at the time of death. The extracted DNA was then hybridized with RNA baits designed to match the modern Yersinia pestis genome. These baits effectively fished out the rare ancient plague DNA from the soup of contaminants. 7 individuals from the 1338 to 1339 graves were screened. 3 tested positive for Yersinia pestis. 2 yielded high-quality genomes, designated BSK001 and BSK003. It was plague. The tombstones had been telling the truth, but testing positive for plague only confirmed what the inscriptions already said. The real question remained, was this the plague, the one that seeded the entire pandemic, or just one local outbreak among many? To understand what the researchers found next, you need to understand something called the Big Bang of plague evolution. Previous genomic studies of Yersinia pestis had identified a massive diversification event, a moment when a single ancestral strain suddenly split into 4 major branches. It's called a polytomy, and it looks like a star-shaped radiation on the evolutionary tree. Branch 1 became the lineage responsible for the Black Death in Europe and subsequent outbreaks in Africa and the Americas. Branch 2 spread across medieval Eurasia. Branches 3 and 4 are currently circulating in rodent reservoirs in China and Mongolia. Scientists knew this Big Bang happened. They could see it in the genetic data, but they couldn't locate the ancestral node, the original strain that gave birth to all 4 branches in time or space. It floated theoretically somewhere between the 10th and 14th centuries. The question was, where and when exactly did this diversification happen? When the team analyzed the phylogenetic position of the Chui Valley strains, they found something that ended the debate permanently. BSK001 and BSK003 were identical to each other, and they sat at the exact node of the Big Bang. They weren't descendants of the mother strain. They were the mother strain. The implications are absolute. The strain found in the 1338 graves is the direct ancestor of the Black Death strain that ravaged Europe 8 years later. It's the mother from which the European, African, and East Asian branches diverged. The specific dating of the tombstones allows the Big Bang to be dated with unprecedented precision, the first half of the 14th century, centered around 1338. And here's the detail that demolishes the China hypothesis. Branches 3 and 4, the modern strains found in China, are genetically downstream from the Chui Valley node. They're descendants, not ancestors. The plague didn't move from China to Central Asia. It moved from Central Asia into China. To determine whether the 1338 strain evolved locally or was imported, the researchers compared the ancient genomes to modern Yersinia pestis strains circulating in wild rodent populations today. The match was precise. Modern strains found in gray marmots in the Tian Shan Mountains, the range surrounding the Chui Valley, are the closest living relatives to the ancient BSK strain. The ancestors of the Black Death didn't travel thousands of miles to reach Kyrgyzstan. It evolved in the neighboring mountains, spilled over into the human population, and then radiated outward. Gray marmots, Marmota bibecina, are large, social rodents that live in high-altitude meadows. For millennia, Central Asian populations hunted them for food, fur, and leather. The flea vector, Oropsilla silentiwi, can survive in marmot burrows even through harsh winters. In a stable ecosystem, the plague cycle remains sylvatic, confined to animals. But spillover occurs when ecological barriers are breached. The emergence of the plague in 1338 correlates with significant climatic fluctuations. Paleoenvironmental data suggests the region experienced a wet period in the years before the outbreak, followed by instability. Increased precipitation boosts vegetation. More vegetation supports a population explosion among marmots. When the climate shifts back to drought or normal conditions, the inflated rodent population crashes. The fleas, deprived of their primary hosts, seek alternative blood sources: humans, camels, livestock. Phylogenetically, LAI009 is a transitional form. It differs from the ancestral Chui Valley strain by only one mutation, a single nucleotide polymorphism, and it's ancestral to the Black Death strains found in Europe. This is a genetic breadcrumb trail that confirms a westward trajectory. Origin: Chu Valley, 1338. Transit, Golden Horde Territories, Volga Region Intermediate Strain. Destination, Black Sea, Crimea, 1346. This trajectory aligns perfectly with the movement of trade caravans and the military expansions of the Golden Horde. The fur trade likely played a critical role. Marmot pelts carrying infected fleas could transport viable bacteria over thousands of kilometers. The traditional narrative of the Black Death's entry into Europe centers on the siege of Kaffa in 1346, where the Mongol army of Jani Beg allegedly catapulted plague-infected corpses over the city walls. Whether biological warfare actually occurred remains debated. Infected rodents entering the city are a more epidemiologically plausible vector than catapulted cadavers. But the genetic evidence confirms the directionality. The plague did arrive from the east, specifically from the Golden Horde territories, where the virus had been percolating since its emergence in the Tian Shan. The Genoese merchants fleeing Kaffa in 1347 carried direct descendants of the Karadzic strain to Constantinople, Messina, and Marseille. The medieval sources weren't wrong. They just lacked the precision that only genetics could provide. So where does this leave the three competing hypotheses? The Central Asian origin is conclusively proven. Direct genomic evidence, dated archaeological context, and phylogenetic positioning all converge. This isn't circumstantial. It's molecular fact. The China hypothesis is genetically disproven. The modern Chinese strains descend from the Central Asian node, not the reverse. The 1331 epidemic in Hebei may have been a different disease, or if it was plague, it represents a secondary spread from the Central Asian hub rather than the primary source. Critically, there is zero archaeological DNA evidence from 14th century Chinese sites. The steppe hypothesis is confirmed, but not as an origin. The Golden Horde territories acted as a transit zone, a bridge where the pathogen moved and evolved slightly during its journey from Central Asia to the Black Sea. The steppe was the highway, not the factory. Even with this breakthrough, mysteries linger. Why did the spillover happen in 1338 specifically, and not earlier? Marmots had been in those mountains for millennia. Humans had been hunting them for just as long. What made that year different? Was it purely the climate anomaly? Hunting pressure? A random mutation in the pathogen that increased transmissibility? Or a confluence of factors that created a perfect storm? Could there have been earlier, undetected spillovers that didn't spread beyond local populations? Future discoveries, more ancient samples from the transmission route, paleoclimate data with finer resolution, additional genomes from marmot populations could refine the picture further. But the core question has been answered. The resolution of this mystery required a convergence of disciplines that were, until recently, methodologically incompatible. Epigraphy and history provided the date scholars like Philip Slavin reading Syriac inscriptions and analyzing 19th century excavation diary. Archaeology provided the samples cross-referencing museum catalogs to locate specific skeleton. Genetics provided the proof. Maria Spyrou, Johannes Krauss, and their team reconstructing ancient genomes and mapping phylogenetic relationships. Ecology provided the mechanism understanding marmot reservoirs, flea vectors, and climate triggers. No single discipline could have solved the puzzle alone. The mystery required consilience, the unity of knowledge. For centuries, the story of the Black Death focused on the arrival of ships in Messina, treating the East as a vague, monolithic backdrop. The 2022 discovery shifts that perspective permanently. Central Asia wasn't a nameless source. It was the epicenter. The Chagatai Khanate, the Silk Road trading communities, the cosmopolitan mix of Nestorian Christians, Mongol rulers, and local populations. They were at the center of this story, not the periphery. It validates the accounts of non-European medieval sources and places Central Asia at the heart of global health history. The Big Bang of 1338 serves as a historical case study for pandemic emergence. It demonstrates that massive global health crises often begin with subtle, localized spillover events driven by environmental changes and human-animal interactions. The chatter in the Chui Valley, a localized outbreak in a trading town, was the precursor to a global catastrophe. Understanding these pre-pandemic signals in wildlife reservoirs remains as critical today for emerging zoonoses as it was in the 14th century. The mystery that haunted historians for 7 centuries has been solved not by ancient texts or educated guesses, but by the molecular record locked inside teeth buried beneath a cemetery in the mountains. The Black Death didn't emerge from a vague, mythical East. It came from a specific place, at a specific time, for specific ecological reasons. 1338. The Chui Valley, Kyrgyzstan. The Tian Shan Mountains, a zoonotic spillover from gray marmots, triggered by climate, amplified by trade, and carried across continents by the very networks that connected the medieval world. The land of darkness has been illuminated, and the answer was hiding in plain sight, carved into tombstones, waiting for science to catch up to history. Here's my question for you. If you had to bet your reputation on where the Black Death originated, based only on historical records before DNA evidence, which hypothesis would you have chosen? China, the steppes, or Central Asia? Drop your answer in the comments and explain your reasoning. If you want to see how ancient DNA is rewriting other major historical mysteries, the next video breaks down the genetic evidence that's forcing us to completely rethink the peopling of the Americas, and it's way stranger than the plague story. Hit subscribe so you don't miss it.